Welcome to the CanMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amaralt. I'm the marketing manager at Medicinal Genomics and proud member of the team that puts on the CanMed conference. On this episode, I had the pleasure of speaking with Dr. Zamir Punja. Zamir is a professor of plant biotechnology at Simon Fraser University in Canada. His research interests include the etiology and management of plant diseases on vegetable and horticultural crops, and the applications of biotechnology for disease management. Since 2018, his work has shifted to researching on cannabis, where his group has described a range of previously unreported pathogens affecting the crop and has evaluated various methods of disease management. Zamir's latest research has been focused on a pathogen that is spreading throughout cannabis cultivation facilities worldwide, hop latent viroid. HLVd, as it's also called, is a relatively new pathogen. It was first identified in hops in the late 80s and was just detected in cannabis in 2018. It is also quite small, only 40 nanometers in size. Still, in just 5 years, this tiny pathogen has managed to infect as much as 90% of cannabis facilities in California alone, all while reducing yields as much as 50% and costing the industry $4 billion in losses. Zamir shared his latest research on HLVd at CanMed 23, which is the basis of our conversation today. I've put a link to his video presentation in the show description. Our conversation covers why Zamir considers HLVd to be the COVID of cannabis, how HLVd spreads, the effect HLVd has on cannabis plants, including some really amazing electron microscope images that he captured of affected trichomes, practical recommendations for testing plants for HLVd, and possible solutions to the HLVd problem. Before we get to my conversation with Zamir, I want to thank this episode's sponsor, Pheno Express. Pheno Express offers low-cost genetic testing services to cultivation facilities. Using qPCR technology, Pheno Express helps cultivators identify genetic traits, such as plant sex, while plants are still in the seedling tray. They can also identify plant pathogens, such as hop latent viroid, lettuce sclerosis virus, and powdery mildew, before the plants show obvious signs of infection. Contact Pheno Express today so they can help you predict, prevent, and eliminate major crop problems before they happen. Learn more at phenoexpress.com. That's phenoexpress.com. Okay, and without any further ado, please enjoy my conversation with Dr. Zamir Punja. Good afternoon, Zamir. Thank you so much for joining us on the podcast again.
Thanks, Ben. Good to see you.
Excellent. I'm excited to talk with you today about a topic that everyone seems to be talking about, and that's hop latent viroid. If you're watching or listening and you're not familiar with HLVd, well, you're in the right place because Zamir has done extensive research into the viroid, much of which he shared at CanMed 23 in his presentation. I'll put the link to that video in the show description so you can check that out. But today, not only are we going to cover what HLVd is, but also talk about ways to potentially prevent and or eliminate it. So let's get into it. Now, Zamir, at CanMed 23, you called hop latent viroid the COVID of cannabis, which does have a nice ring to it. And I think it's a good way to describe what is going on in the cannabis industry for those who might not be familiar. So please explain why you believe HLVd to be the COVID of cannabis.
Well, I thought I'd throw that in because there's a lot of similarities, between what we've experienced with COVID over the last 3 years and what's happening with the cannabis industry. First of all, these are both organisms that one's a virus, which is the COVID, and the other one's a viroid. but they behave very similarly. All the literature shows that viruses and viroids behave in a very similar manner. So that was one obvious thing that was draw my attention to the fact that there are many similarities. The other being it spreads really easily by hand. And we know with COVID, wash your hands, make sure you don't touch surfaces, and when you blow your nose, make sure you throw away the tissues, and so on and so forth. So any material that comes into contact with your hands that has hop latent infected material like sap is going to transmit it. So that was another analogy that I thought was pretty interesting. And also it spreads really well, both COVID and the hop latent. Now, I should make a distinction here that hop latent does not spread in the air. I've seen some reports where people say, "Oh yeah, it's airborne. You got to watch your vents." It doesn't spread in the air. Now, it'll spread through seed, obviously, and if there's pollen, the pollen will fertilize. But in a general operation where you're growing plants, mothers, flowers, you don't have to worry about the airborne aspect of hop latent. Now, unlike COVID, of course, where we develop vaccines, we don't have vaccines for hop latent. We've got to develop some other methods to address it. And one of the other main differences, which I think is important, in COVID, as you remember, we were getting a lot of mutations, right? New strains were coming up all the time, and they had to develop new vaccines for that. Right now with hop latent, we haven't seen a lot of genetic diversity. In other words, it looks like the first report in California, that genetic strain that was there is very similar to one in Massachusetts, is similar to the one in Colorado. We've actually found it in Canada. So it almost looks like right now, anyway, it's genetically, fairly stable. It's not diverged itself as much. What that's due to, maybe with more selection pressure, in the human population, there's so much diversity, people react differently. And so the virus is constantly trying to keep up. Maybe in the cannabis world, it's perhaps not the same. The other thing with COVID and hop latent, the symptoms, right? You've had people that are asymptomless with COVID.
Right.
They test positive, but no fever, no runny nose, no sneezing. And in cannabis, it's the same thing. You have these asymptomatic plants that, unless you do a PCR test, which we'll get into later, you may or may not know that you've got hop latent because the plants may not show any symptoms. So there's a ton of similarities between the two types of diseases. I just wish we had the kind of money that was thrown at COVID, thrown at us in cannabis, because I tell you, if we did that, we'd be done in a year. Like, we'd have all the problems solved, because you need money for research, and that's getting pretty tight.
Yeah, for sure. And there were a few things that you mentioned in there that I jotted down that might be kind of news to me, because I know that there has been some discussion and debate about whether HLVd actually spreads through seed. So it seemed like you were just saying that yes, it does. So
yeah, there's some pretty good evidence that Toomey Genomics released and shared with us, as well as work that they did with Texas A&M. And they did two things. They took infected mothers and added pollen that was clean from a clean plant, and they found that a high proportion of seeds that came out of that had hop latent. And we'll talk about on the outside versus the inside.
Right.
And then they took a healthy mother, and then they took pollen from an infected male and put that onto the plant. And again, they got seeds that had hop latent. The infected females that received healthy male pollen had a higher seed infection frequency. So the mothers being infected is more important than the father being infected, or the pollen. And a lot of the contamination or hop latent was on the outside, on the seed coat, which is very common with many viruses. But they did show maybe 10-15% infection inside the seed, where they did experiments where they totally cleaned the outside of the seed to the point where the seed was pretty much killed. And then they rescued the tissues from inside and ran a PCR on it, and it was positive. So it's both outside and inside, and infected mothers play a more important role than infected pollen.
So now, if you did have a seed that had the viroid on the outside and maybe not in the inside, but you didn't clean it, disinfect it, or whatever, and were to plant that seed, could it infect the plant that emerges?
It could, in cases where the seed coat, was still attached to the cotyledon coming out. Right. So you've got the plant that's growing, and there's some abrasion against the cotyledon. You could get transmission there through mechanical damage. Um, and really the only way to know for sure, obviously, would be to test that seedling. So, you've got 5 seeds, really, high-priced seeds, and you've grown them up and you've got these 5 seedlings. I'd test every one of them just to be sure, because that's a great point where hop latent can start, and you want to eliminate that option. So testing seedlings, we'll talk about what the right stage of testing would be, but I would say the best way is really just to test to make sure.
Excellent. And just to kind of complete, I guess, setting the stage for, people who might not be familiar with hop latent viroid, we talked about, how it's spreading and its similarities to COVID, but what is it actually doing to the plant? How, what are the symptoms? Sort of, why is this such an issue with cultivators?
Right. So, when the first discovery of hop latent was made, it was made in hops. And this was back in about 88. And so the researchers figured out what the sequence of the viroid was. And they didn't see any symptoms on the plant. And when they tried to transmit it to other plants, they didn't see anything happen. So they gave it the name hop latent. So the word latent basically means no symptoms. And again, with COVID, you could be infected but be latent, right? You're harboring the virus, it's in you, but the PCR tests are showing it's there, but you're not expressing symptoms. So that's where the latent comes in. But then later, at some point, and usually it's when the plants go into flower, that we see the most dramatic symptoms. That's where I first recognized it, when I looked at a flowering plant, and it was much shorter. The flowers weren't developed as well. The budding symptom, the flowers were smaller. And these are the same symptoms that the California researchers in the 2 different labs reported. That's how they first saw hop latent viroid, was on these flowering plants. And then when you measure THC and CBD and terpenes and so on, what you find out is that they've dropped. They've dropped, depending on the genetics of the plant and when the plant got infected, anywhere from 10 to 40% lower. And that's when the alarm bells start going off, right? You've got this infection that came literally out of nowhere because they hadn't seen it before, but it showed up on the flowering plants. Now, when you work backwards, you can see symptoms in mothers. You can get mother plants that are somewhat stunted. You can see symptoms in the veg, particularly depending on the susceptibility of the genotype you're working with. I've seen stunted plants in veg that are obviously hop latent infected. The leaves are slightly curled. But I wouldn't rely on symptomology alone. I would definitely do the testing to make sure. But the alarm bells go off when you're walking through a flowering room and you're looking at the plants and you recognize that genotype, but yet you're looking at it and saying, these flowers haven't developed. They're smaller. They look like they're like 2 weeks behind. That's when you may understand that you've got hop latent.
Excellent. So now you mentioned hops, and obviously it's in the name there, so it makes sense that it started there. But did I hear that right, where hop latent viroid never really had a negative effect on hops, like any variety?
Yeah. So when they first started, they didn't see an effect. But then, like as research shows, the more you look into it, the more you find. Other labs started looking at it, particularly in Europe, in Germany and other countries, and they started to see, and in the UK, they started to see the cones, the flowers, the end result of the flowers in hops are called cones. The cones had lower acids and phenolics and other chemicals, that essential oils and terpenes that are used to help enhance the flavor of beer and so on. They were starting to see lower quantities of that. The plant itself was not affected. If you've ever seen a hop plant, that thing, it grows in front of your eyes. I mean, it's putting on 12, 14 inches of growth a day. So it's unlikely hop latent does much on that, but it did seem to affect the flowers. And that, when I read that report, I thought, okay, if this is happening in hops, it's got to happen in cannabis, because they both have similar flowering structures and so on. And so now they're finding, but it's very genotype dependent. Depending on the cultivar, some of them seem to be fine. They resist the virus or the viroid, and others really come down hard. And again, I draw the analogy to COVID. Some people get it and they're walking away fine, and others have to be sent to the ER. And a part of it is what your genetic constitution or background is that makes the difference.
Yeah, and so has that been the way that the hop industry has sort of dealt with the issue? They've sort of just moved towards using cultivars that are more tolerant of it?
They've done that, and they're aware that it's spread by mechanical transmission. So in hops, where they use machines to maybe prune the plants or they take cuttings, they're very careful to know that it's spread that way through the sap, which is useful information because the exact same thing happens in cannabis. When you take cuttings and you make vegetative plants out of the original plant, or you're using shears or tools that weren't necessarily cleaned properly, that will spread hop latent, and that's what they found in hops as well. They didn't see very high transmission in seed. It was there, maybe 5 to 8%, not as high as what we're seeing in cannabis, but it may be that the female plant, the mother plant, wasn't as heavily infected, and so there was a little bit of infection, but it wasn't enough to create a very high seed transmission rate as we see in cannabis.
Yeah, and you mentioned too that when you were explaining some of the differences between HLVd and COVID, that the number of different variants didn't seem to be apparent in HLVd. Now, when the viroid jumped from hops to cannabis, was there a change there, or is it effectively the same viroid?
Yeah, it's a good question. The sequences that we have and the ones that Kevin McKernan has looked at medicinal genomics, they're identical to the 1988 report on hops. And so there doesn't seem to have been a significant amount of change, at least in the genetic structure. Now, some of the papers I read on other viroids, there's about 32 other viroids in other plants, like potato spindle tuber viroid. They see a lot of genetic diversity, and part of it is when the viroid is moving from host to host. So potato spindle tuber viroid has a big host range, tobacco, tomato, petunia, lots of plants. And they were able to show as it moves into these other hosts, I guess it needs to adapt itself to this new environment and creates a new strain or a new variant. And that could be what's happening in humans, right? When it jumps from China to India to America, it's mutating as it goes through. We all have different genetic backgrounds, genetic composition, and we're somehow selecting more variants in COVID. So far, knock on wood, we haven't seen that with hop latent.
Yeah, because could that be more of a problem if it were to start doing that?
It would, because all of a sudden you think you've got this genetic strain that's resistant, and now you've got a new variant that is more aggressive. Again, very similar to what we see in COVID, and that's where these vaccine companies are making a fortune. Because this year again, I already got my little reminder that I've got to go for my whatever it is, 4th or 5th vaccine booster. They're going to develop another one, a different booster for COVID, because they say the strain has changed since last year. So they're continuously trying to keep up with this evolution.
So you mentioned too, so HLVd, it seems to be a relatively new phenomenon that we're seeing here. But I wonder, because you've studied a lot of different pathogens in cannabis, has it been around longer than maybe we know of and we just were not aware of it? Like, were you looking at plants and kind of expecting to find one pathogen and not finding it and being like, this doesn't make sense? And now that you know about HLVd, now the picture is clear.
Yeah, it's one of those things, the more you look, the more you find, right? But prior to 2018, which is when I started my work, there were no reports of hop latent. And part of it was because it hadn't been discovered, it hadn't been tested. And hence the 2 reports out of California in 2019. They published in 2019, but the plants were sampled back in 2018. I think it's been around. I can't say how long. I would guess maybe 5 to 8 years. Somehow it migrated over from hops. Clearly, somebody was working with hops and maybe had sap on their fingers, or something happened where it was then moved over into a cannabis production facility. But it's probably been around, I don't know, 5 to 8 years. And because it hasn't been recognized, we weren't really looking for it. If I saw a stunted plant, I would look, oh, that's probably Fusarium or Pythium, not even knowing what hop latent is. When they first mentioned it to me, 2018, I go, don't worry about it. It only affects hops. It's latent. And that's a mistake that everybody makes initially. And then we realize, my goodness, this organism is very well adapted to infect cannabis and cause damage.
Right. And now, part of your presentation at CanMed too, you shared some really kind of stunning images that you captured with an electron microscope, and with regular photography too. But I think the ones in particular where you're looking at the trichomes are cool to look at. And if I'm going to try to share my screen now, we could kind of take a look at those and show the folks, if you bear with me for 1 second.
Yeah. So I guess while we're waiting, I can explain. The scanning electron microscope is a very, very powerful piece of equipment that magnifies things tremendously in the magnitude of thousands. And so when you look at trichomes, as we see here now, you can see these at this magnification. You could probably see this with a high magnified hand lens, or even under a dissecting microscope. So even though this is under a scanning, this kind of thing I think you can see with a normal microscopes. On the left side there, you've got the normal stalks and the large glands or bulbs at the top of the stalks, and over on the right you can see that they're smaller. They're there, but they're smaller, and they look like they're not as well developed. Now the numbers are the same. This is what surprised me as well. When you look at numbers, you actually sit and count these little dots. They're equal numbers on both sides. So when people say, oh, well, the trichome numbers are down, they're not actually down. They haven't developed as well. They're underdeveloped because of this infection.
Yeah, and of course, the trichomes themselves, like that's where the plant produces the cannabinoids and terpenes and everything, correct?
Yeah, I mean, this is why the industry is doing what it's doing, is to collect all of the cannabinoids and THC and other things that are called resin from these glands. And so anything that disrupts the development of these glands or makes them smaller or a poor quality, then that's going to affect the ultimate amount of THC and CBD and so on that you have. So when one looks at that, it's basically the plant is under some kind of stress, right? It's supporting the reproduction of this viroid, which, by the way, it's producing millions and millions of copies of itself in the cell, in the plant cell. It has no other way of replicating. Again, similar to COVID. COVID has mechanisms to replicate in humans, but it still takes a toll, right? People that suffer from an infection with COVID are in bed for 6 or 8 days. They're weak. They've lost appetite and so on. In the plant, this thing is taking energy out of the plant by making millions of copies. And so partly I think it's the stress level on the plant. And partly it may also be that the viroid is actually shutting down parts of that pathway that produce these cannabinoids. We don't know for sure. There's a lot of research labs doing this kind of study right now to find out what exactly is being turned off in these infected plants. And I bet you a lot of the genes that are involved in cannabinoid synthesis are also turned off or downregulated, which would also explain why we're seeing this reduced cannabinoids. Now, it's not just cannabinoids. We see plants turning yellow, which means they're lower in chlorophyll. We see plants that are stunted, which means there's less growth in the apical meristem. So there's other things that happen in these plants. So there's a really good example of a particular strain or genotype that is very distinct. I look at that and go, boy, that's hop latent. Sure enough, every one of those is infected. And you can sort of see fewer trichomes on those surrounding leaves or bracts. And then you've got less chlorophyll, which is, again, another effect of this viroid.
Right. No, and I did want to get to these, because I think this is some of the most striking images. I mean, that looks like a deflated balloon. Yeah. And
this is, I really was happy to get this, or at least the graduate student and I that worked on this were really happy to see this. There's nothing more visual. They say a picture paints 1,000 words. There's nothing more visual than showing a grower or a producer or a regulator. This is what hop latent does. On your left is the healthy, plumped up, full of cannabinoids. On the right, it's deflated. It most likely inflated first. So the way these trichomes develop is that the outside, the gland itself, the head, is inflated first. So it's ready to go. And then it's the cannabinoids start to fill it up. But if you've got a situation where the cannabinoids are not filling it up, that it's 40% less, then this thing will start to collapse, right? There's no oxygen, or there's no support within those so-called hot air balloons. I use the term hot air balloon to describe this. So these are dried flowers now. And so on the left, you can see the drying does not impact the structure of the trichome gland or the head. It's intact because the cuticle is really strong. But on the right, where you've got hop latent infected, because it hasn't inflated completely, it's getting to be like a flat tire. There's no air in there to begin with, but then when you start drying these trichomes down, you see this shriveling effect. It's quite dramatic.
Yeah, so that's really cool, because when I first saw this, I thought that, well, I mean, this is indicative that if you have a shriveled up trichome, it's not going to be producing as many cannabinoids. But what you're saying is that this is just showing that it hasn't been filled with those actual cannabinoids. So that's interesting. Wow.
Yeah, because the plant's going to go ahead and create this cavity, in order to prepare itself to produce the cannabinoids. And usually the cannabinoid production starts when the stalk, which is the bottom, starts to elongate, and that's been shown by previous studies. So when the stalk elongates, that's when this cavity starts to fill up. But now you've got a hop latent infected plant. It's not producing as much cannabinoids, and so it doesn't fill up.
Yeah.
And then when you dry it, you get this effect. Sort of, you're looking at a plump grape on the left and a raisin on the right.
Wow. Yeah, no, and these are fantastic images for the very fact that we were talking about, like it's latent. It's not always obvious if you were looking at a plant that it's got any issues with it. Maybe not until you take it to the lab and get it tested. But if you zoom in like you have, well, here it is.
Yeah.
So I think now we can kind of transition into talking about, well, we've established the problem. What do we do about it? So if you were a grower and you suspect that you have an infected plant, what are your options?
Right. So there's many things growers can do. I mean, the first thing they need to do for sure is confirm that they've got hop latent. So whatever plant they're dealing with, most likely it's a flowering plant, because that's usually when you see it and they send it off for testing. The first thing would be to work backwards and try to figure out where did I get my clones from for that particular plant or that particular genotype. Or did I grow it indoors? Did I purchase it from someone else, or did I start it from seed? And once you've sort of worked backwards, you can say to yourself, all right, let's assume it came from clones within your own facility. That means the mother plant definitely has hop latent. So you work backwards, you then test your mother plants, depending on how big they are and so on. We can talk about sampling strategies, but you test your mother plant. And obviously, if the mother plant is infected, the bad news is that I would say it's got to be thrown away. You've got to get rid of it. There's really no proven ways right now to rescue a plant that has hop latent in it. I mean, we've talked about meristem culture and tissue culture and so on. It's possible, but it hasn't been demonstrated on a high level, on a high scale, that you can rescue a mother plant that's got hop latent. So if you've proven that it's your mother plant, then obviously you get rid of that. The other possibility is that it picked it up when the plant was in veg. So let's say you're vegging on the table, and if you're using hydroponics, then you've got water or nutrient solution moving between plants. We've shown, and others have shown, that it can move with water, with recirculating water. And so a veg plant that's maybe 2 weeks old could potentially have picked up hop latent through the root system, and the viroid then makes its way into the plant that way. And then, of course, the last alternative is a seed. If you started from seed, you didn't test the plant that came off the seed, it could be that gave rise to your infected flowering plant. So it's a detective, sort of a detective process, or a process of elimination, deciding where did it come from. You need to know that. That's number one.
Yeah, and I think you kind of answered my next question, but I think it's a good thing to hammer home. There really doesn't seem to be any sort of treatment or kind of remediation options available yet, right?
Yeah, if you've got a heavily infected or even an infected mother plant. Now, people have tried, and people are trying, taking cuttings from different parts of the big plant. So you could definitely do that. You could take them from the top, the actively growing. You could take it from the side, the bottom, but then you have to go through and test all of those to see if by chance, and really it is chance, if by chance you were able to get a clone that was free of hop latent. When the viroid gets in, and we can discuss this now, the first place it goes is down into the roots. Right. So let's say I infected it through a cutting. I went with my tools and I transferred some sap onto a cut surface. And the first place it goes is down into the roots. And that takes about 2 weeks. So let's say 2 weeks after it was introduced, it goes down into the roots. And then from the roots, it's moved upward. And the reason it's moved upward is it sort of follows the flow of sugars that are being produced by photosynthesis in the plant. And that usually is towards the actively growing shoots, which are the young shoots. So you'll find it in the young shoots probably around 4 weeks after that initial infection occurred. And then it starts moving around the plant. So let's say by 6 weeks, it's now pretty much everywhere. But there are cases where, a branch gets missed. You've got a diseased plant and all of a sudden, a healthy branch on one side. And I think I have a photo in that presentation. We can come back to it in a second. For some reason, that branch got missed. If you're lucky enough, you can get a cutting out of that, a clone out of that, and you may have escaped hop latent, at least in that particular plant. So it's not a uniform distribution throughout. And so some producers will go through and take clones from various parts of the mother plant in the hopes that they can find a few that don't have the viroid.
And that is effective?
I don't know. I don't know. I mean, it obviously, they all have to be tested.
Right.
They all have to be tested because you don't know for sure whether you lucked out or whether it's dormant, like it's there, but it hasn't had a chance to grow out and replicate. Now, meristem culture, which is taking the very, very, very small tip, does seem to work in many other plants. So in potato, particularly here in Canada, when we had an outbreak of potato spindle tuber viroid back in the 80s, the government stepped in and said, nobody's going to be able to grow any potatoes unless they all came from a tissue cultured meristem plant. And so they started this meristem program throughout Canada, and only growers that purchased these plants that were propagated through tissue culture could grow potatoes. And as a result, it got wiped out. You can hardly find that in most commercial fields because of this rigorous insistence on using clean plants. Now, with cannabis, it's hard to do because A, we have to show that those plants are clean from meristem culture, and B, not everybody's going to want to buy from one source, right? Everybody's got their own preferential source for plants. And so there's no guarantee that the plants you buy will be free of hop latent unless they're tested.
All right. So yeah, that's a good segue. So let's talk about testing, because one of the things that you mentioned in your presentation is that sort of the only real way to get rid of an infection, if you have it in your facility, is sort of this systematic testing and removing of infected plants. So I'm wondering, sort of what is the protocol for that? Which plants should be tested, and is it all of them, just the ones that sort of appear sick, just the ones that are kind of in the same vicinity as some confirmed positives? What are sort of the best practices in developing that testing protocol?
Yeah, so definitely a systematic approach has to be taken, and one where the grower is taking a lot of records, so they can follow through what exactly happened from A to B to C. The first place definitely starting with the mother plants and testing those to see if they're clean and whether they may accidentally have picked up hop latent viroid. I would say the second place to test would be your veg plants. Somehow, if some viroid got through and wasn't detected in the mothers, you may see symptoms in that veg plant. Sometimes they are a little bit shorter, may have slightly curled leaves. So again, being very vigilant about what the plants are supposed to look like. And most growers have had years of experience. They may look at that plant and goes, that's not growing right. Did I not fertilize it properly? Is it not getting enough light? It may actually be infected with hop latent. I would not test necessarily flowering plants, only if you want to make sure that you've got it or don't have it, because at that stage, it's too late. Right. There's not a whole lot you can gain from testing a flowering plant. You're going to get rid of it anyway. It's mostly the mothers and the veg plants. Now, clones, when you test clones, they should be rooted. So if you get a bunch of unrooted clones from someone and you want to get it tested for hop latent, I would suggest rooting them first, because if they are infected, the first place the viroid's going to go is back down into the roots. And we've shown when you've got a 2-week-old rooted cutting, that if it came from a mother plant, you will find it in the roots 100%. You may not find it in the leaves that may originally have had the viroid, and you may have tested it and found a negative. I would say root it, get the roots coming out, take little samples off those, send them off. If they come back negative, it's negative. If it comes back positive, throw it out.
Wow.
Okay, so, and a lot of people do import a lot of clones, and most of them tend to be not rooted. Root them first, and then test them, and then get rid of the ones that turn out positive. The beauty about the root sampling is it's more sensitive at that early stage, because the roots are starting to accumulate that viroid. Later on, when that plant grows up, so let's say it's now 6 weeks old, you can find the viroid everywhere in the leaves, right? We talked about how it takes 2 weeks to go down, 4 weeks, and then 6 weeks. So there's a recent paper that came out that basically says you can test leaves. It's not a problem. Only if that plant is old enough, it's 6 weeks, 8 weeks, 10 weeks old, and it's had a chance to spread the viroid in the leaves. If it's an early plant, a clone or a veg plant, go for the roots, because that's where you'll very likely find it, because it hasn't had a chance to move out into the foliage as yet.
Right. And then I guess further complicating things, because I know that some experiments that we've done or our team has done, we've seen plants where it's detectable in the roots, but never actually makes it up into the upper leaves, which could be, could be tolerance that we're looking for. But yes, like you said, if it's in a hydroponic system and those roots are in the water and that water is circulating, well, there you go. Now you have your asymptomatic spreader, right?
Yes. And so you're bang on in terms of because it does go down to the roots. We can use that as a consistent method for testing. However, it's not to say that it's necessarily going to move into the leaves. Now, everything we work with is highly susceptible. So I've never seen a situation yet where it's in the roots, but eventually doesn't find its way into the leaves. So that, you're right, it could be a tolerance or a resistance, which makes sense. The plant is fighting it. It's keeping it localized, doesn't want it to spread up into the leaves and flowers. But, environment plays a pretty important role in terms of how it moves from the roots into the leaves. So we find, for example, when you flip or when you turn the lights into 12 hours of light and 12 hours of dark, that plant that only has viroid in the roots, all of a sudden will start showing it really fast in the leaves and the flowers. So again, it may be that flow of sugar that's being triggered by the flipping of the lights that's causing this movement back into the upper leaves. So I'm doing some experiments now where I'm putting plants in the darkness, I'm putting plants in full light, I'm putting plants at different temperatures, trying to see if I have a root infection, what's it going to take to move that viroid up into the upper part of the plant. Because maybe we can play around with environment. Environmental conditions might reduce the rate of spread, as well as genetics.
Yeah, because I would think particularly in the cases of a mother plant, right, where if you're taking cuttings from the canopy, from the leaves, even if it were to have an infection in the roots that never got to those leaves, could produce clean clones.
Yes.
Theoretically.
Yes.
But I do want to go back to sort of like that testing protocol you said, where, you said start with the moms and to test them. So what are we looking at for sampling? Like, I guess it's kind of a good point. Should you be testing the roots or the actual branches that you're going to be taking cuttings from? Everything? What do you recommend?
So right up until this past year, the entire industry was sampling leaves.
Yeah.
And it worked. It worked. There's nothing wrong with what they were doing all these years. All the testing labs that I know of were either taking leaves or petioles. Petioles seem to be a little better. Petioles are the connections that the leaves have to the stem. Because again, it's the vascular system, right? It allows the movement of the sugar in there. And they were finding hop latent. All the reports that we have pretty much are based on leaf testing. So a big mother plant, they're saying 4 samples, either 4 samples through various parts of the plant, either separate or you can combine them. And that'll give you one composite sample and maybe save you a bit of money because you're not testing individual samples. You're combining the tissues into one tube. The root system on one of these larger moms is harder to get to. because really the ideal sampling location is the root system more towards the central stem. And so digging out in there and causing some potential damage may not be worth the effort. So on a larger mom, and I'm going to say 2 months and older, you're probably okay with taking these 4 leaf samples or petiole samples and combining them. Anything younger than that, where it's still potentially in the roots and maybe hasn't spread uniformly, I would go with the roots. And so that would definitely be clones. It would definitely be veg plants, and potentially plants that you're trying to save for future mums that are 6, maybe 8 weeks old. Go for the roots. On an established mother that is in your nursery, you've had for maybe 3 years. I don't know how long growers keep these plants. The leaves will probably do a good job if your sampling is systematically done throughout the canopy.
And how frequently? I mean, should you be testing monthly or just when you're taking cuttings? What's your advice there?
Yeah, that's a good question, and I guess it's going to depend on whether you've got the same moms that you're using over and over. In which case, I wouldn't really sample them more than once a month. If it's an established mother, if it's a younger vegetative plant that grows rapidly and you're just about ready to take it into flower, I would test it again after 2 weeks from the first test, just to see whether you may have missed something and it's outgrown itself since. So it's going to vary from 2 weeks to 4 weeks. I know some growers like to sample every week just to be absolutely sure that they haven't missed a potential positive, because I guess if you missed it and that's going to go into the flower room, you've lost a lot of potential revenue by not having tested that one plant, as opposed to paying whatever it is, $50 for an extra sample. And so they may do it every week, but I think every 2 weeks on actively growing plants, and every 4 weeks or once a month on plants that are larger and perhaps not growing as rapidly.
Excellent. And that would be, and sorry, say again, which ones would be roots, which ones would be leaves?
So definitely the younger plants. So anything I would say younger than about 6 weeks, do the leaves. Do the roots.
Right.
Because they're still growing. They're still developing root systems. They're still trying to move the viroid. If they're plants that are older, 3 months and older, the leaves should be fine. As long as there are multiple samples, right? At least 4 that are combined or sent in separately. And similarly for the roots, we find if you take 3 or 4 small root samples and combine them into one, that one result is good enough to tell you whether the plant's infected or not.
And now, is this same protocol applicable if you have a known infection or not? Like if you know you've got infected plants, do you up your testing or what?
Well, technically, that infected plant shouldn't be in your facility, but if you've decided to keep it, then definitely I would test it every couple of weeks.
Well, no, not necessarily if you kept it, but say I have a grow now, it's clean, I'm testing, everything's coming back negative, everything is great. Okay. But now I'm finding, oh no, a few plants over in this corner, now they're infected. Right. Should I do more testing to make sure to kind of mitigate this spread somehow, or? Well,
if it's a definite positive, I would move it out. I would move it into another room and then decide what you want to do. Don't leave it there. Now, it's interesting because I haven't seen any data yet to suggest that the viroid moves very effectively, down the row, for example, if somebody's walking. They say if your clothes brush up against the plant. I don't know that is sufficient damage to the leaf to move hop latent. It does for many viruses and viroids, but there may be data out there, but I haven't seen it yet. And the reason I say that is because I've walked into facilities where they've got a few infected plants here, then everywhere. If this thing was moving with people brushing up against the plants, every damn plant in that location would be infected. And that's not the case. We're finding isolated spots here, there, and everywhere that may have been introduced through a pruning tool. Sap, the movement of sap is super important. So when you brush up against a plant, there really is no sap being released or on your clothes that's going to react on a wound on another plant. But sap on your tools and sap on your gloves. So you've got obvious sap stuck to your gloves and you touch another plant and you damage it, that will transmit it. Damage is the key. The tissue has to be damaged. Usually I think that it has to be, the vascular system has to be exposed. In other words, there has to be a broken vein or a broken off leaf that allows that viroid to enter in. I think just putting the viroid on the surface of a leaf and letting it sit, I don't believe is going to cause an infection. So you got to have the wounding and you've got to have the sap and water. So these plants could have picked it up if there was recirculating water that has the viroid in it and somehow got into root contact. And then, of course, the viroid spread within the plant. So recirculating water is a problem because I haven't seen any treatments yet that work. So even 100 degrees centigrade heating doesn't do it. UV doesn't do it. Fluorination doesn't do it. I don't know yet what treatments could be made to water to clean it up, recirculating water yet. I mean, there's obviously a lot of companies working on that. And eventually we'll have an answer. But right now, you have to be super careful with water that's being recirculated from an infected mother or plant.
Wow. Yeah, that seems that seems like a massive issue. Now, in terms of that, so in addition to testing plants, you should be testing your water and your surfaces, your equipment, things like that as well.
Yeah, so definitely, if you're recirculating and you know you've got a high incidence of hop latent, you're almost guaranteed to have it in the recirculating water. Now, we got to remember it's not pure water that has the viroid. It's little flakes of small pieces of roots and organic matter and debris that's come off the roots, that's sloughed off, that's carrying the viroid in it. And so when the water is recirculated, it's still got a little bit of particles and stuff in it that potentially has the viroid in it. We've seen it on surfaces on, for example, on flood tables. So if there's a plant that's sitting on a table and there's water, up against the roots on the bottom, you can find hop latent on the surface of that table, as well as, of course, on tools. For that, cleaning, bleach does a pretty good job, or Virkon, 2%, because that comes in direct contact with the water or the film of water that's on the table. It can eliminate hop latent viroid quite effectively on tools and on the surfaces of tables. But in terms of what does it do to sap if there's infected sap, I don't know how effective Virkon or bleach are at this stage, where you've got visible sap being moved from plant to plant, whether those two chemicals will completely eradicate the viroid. But for surfaces, there's definitely materials out there that will work.
Excellent. That's good news. Now, in terms of testing, I think you mentioned before PCR, qPCR is the gold standard for testing. Is that the option that you recommend?
I would say so. And most labs realize that, and they've geared their methodologies to test using qPCR. Yeah. There's really no other effective rapid test that will work for hop latent.
Excellent. All right. Well, we covered a lot, Zamir. I want to be definitely mindful of your time. So is there anything we missed?
Don't give up hope. just like everything else, and just like COVID, and there's people obviously that don't necessarily believe in COVID, but I think we've got a good handle on it now. Not to say that it's not going to be a problem later on, but we've got a good handle. We put a lot of good science into it. We threw a lot of money at it, and it's under control. With hop latent, I think it's a similar thing. We put some good science into it, get some money going that can fund research and help companies develop products that will work. Growers in general are very astute. When they know they've got a problem, they'll find every means they can to address it. And I think they're starting to do that. They're starting to see, for example, that certain strains or certain genotypes are more resilient than others. They're going to start using that in their crosses, right, or they're going to start using that to produce. If they know it's in the seed, they're going to be doing seed testing, or they're not going to be buying seed from a location that hasn't necessarily certified it. If they're buying clones, they're going to be a little bit more careful about the sources of their clones. If they've got an outbreak, they're going to do everything they can, including people wearing proper clothes and wearing gloves and sanitizing their tools and so on. And when you use a combination of these things, it's not going to go away, but it's manageable. The more you understand, obviously, the more you can manage. But I know numbers now are scary. 90% of California production has it, 50% in Canada. Those are scary numbers. But I think as we learn more about this thing, in a couple of years, maybe I won't have to do these webinars or podcasts anymore, because it's not going to be important. I mean, 2 years ago, I think you mentioned 2018 when I spoke, hop latent was never on the radar. I mean,
Right.
Fusarium was.
Yep.
Fusarium was big.
Powdery
Now,
mildew.
and powdery mildew and botrytis, which are still big. Now it's hop latent. 2025, hopefully, it's under control.
Excellent. Well, as long as we have people like you doing this work and sharing the information, it's the best we can hope for.
Thank you.
Zamir, before I let you go, if there are any other resources about this that you would like to share, or websites or anything where people can visit to learn more about the work that you're doing, or maybe even just work that you're a fan of that you think would be apropos of the conversation, please share.
Yeah, so I think my latest obviously is the CanMed 23, and I think there's the most recent data is presented there. Definitely, Medicinal Genomics has a good website for a lot of information pertaining to hop latent viroid, as well as Tumi Genomics has a really nice website that you can visit that has a lot of information on management and symptomology and so on. And both of these labs obviously offer testing services, and there's others. There's other labs out there that are obviously getting onto the bandwagon. There's a tremendous amount of information that's being put together on these websites in the last year. I'm really impressed with how much knowledge we have, taken maybe from other systems. And I think the really important part here is the sharing. The sharing of information has been phenomenal. I mean, you guys do a good job of this. Tumi Genomics has done a good job. We're trying to do the best, but we do that anyway, right? At the university, we like to share. But to see these other companies get involved is really gratifying. Nobody's hiding anything. Nobody's keeping things to themselves. They're all out there discussing, and that's the way we're going to make progress. And I think we are making progress. So I'm hopeful.
Yeah,
I may be out of a job in 2 years, but I think there's lots to do.
I think you'll be all right, Zamir. You always have a home at CanMed, that's for sure.
All right.
All right, Zamir. Thanks again for joining us, and hopefully we're going to be doing another one of these soon, talking about total yeast and mold. So folks, stay tuned for that. But until then, thanks again.
Appreciate it. Thanks a lot.
I hope you enjoyed my conversation with Zamir Punja. Check out the links in the show description to learn more about the topics we discussed. And thanks again to this episode's sponsor, Pheno Express. Our next episode drops August 1st, that's 2 weeks from today. In the meantime, please like, comment, and subscribe on whichever platform you use to listen or watch us. Also, please check out canmedevents.com to learn more about our annual Innovation and Investment Summit. You can also view videos of all the past CanMed presentations in the CanMed archive at canmedevents.com. I invite you to join our CanMed community group on Facebook to interact with other cannabis professionals. And lastly, please follow us on social media. Just search for CanMed Events. All right, that's it from us. Stay safe, stay healthy, and be sure to come back for the next CanMed Coffee Talk.